Okay. So this is our project. We chose an article called Well Shark Vision, and this is by Elizabeth, Jocelyn, Stephen, and Camden. And this article just focused on the analysis of the retinal pigments of the whale shark and their role in visual foraging ecology. And it's by Jeffrey I. Fasick and those other people.
Okay. Um, so 1 second. The whale shark is a species of filter feeding shark that feeds primarily on small marine animals, which includes copepods, coral spawn, fish, and squid via 3 methods. That would be surface filter feeding, stationary suction feeding, or subsurface feeding. Each of these 3 methods used by whale sharks occurs within the first 2 meters below the surface level of water. And this is also where broadband light is prevalent. Um, so other animals that forage at this level possess visual pigments that absorb maximally around 500 nanometers. As this is and which is also accepted to be the absorbance at which broadband light is best utilized. It was predicted that the whale sharks would not possess retinal pigments with absorbances shorter than 500 nanometers, as other species who demonstrate visual pigment absorbances values shorter than 500 nanometers often forage at greater depths, suggesting that the shorter absorbances are correlated with the narrow band blue shifted light far below the surface of the ocean. So this would correspond to the biological question: have whale shark retinal pigments adapted to absorb within the broadband light spectrum available at foraging depths, or within the narrow band blue shifted light spectrum? And the corresponding hypothesis that they proposed in this article was that whale sharks do not possess retinal pigments with absorbances shorter than 500 nanometers because they likely have adapted to the broadband light spectrum available at those foraging depths where they get their food. You can go on to the next slide for experimental setup. They used 2 partial whale shark genomes provided by Red and Hara. They were blasted at NCBI, and phylogenetic trees were generated for the respective sequences. And then they used the whale shark rhodopsin, LWS, and melanopsin amino acid sequences to produce 3-dimensional models that were then aligned against either squid or bovine rhodopsin for comparison. And then they also PCR amplified exon 5 of the LWS gene between nucleotides 822 and 984 to confirm the sequence, and observed the melanopsin, which is the OPN4 deactivation kinetics.
Okay, so the main results of the article are listed here. What we really want to focus on is that point number 4, which is that rhodopsin 1 only has a max absorption of 496 nanometers, which tracks with the hypothesis. And then also point number 6, that when compared to mouse OPN4 gene, whale sharks differ significantly in only one nucleotide. And so taking those results and interpreting them, we find that whale sharks cannot absorb higher wavelengths of visible light, which would be closer to the red end of the spectrum. And that makes sense because if you look at point number 3, whale sharks see mostly in blues and a small scale of greens, which are the dominant wavelengths of light near the surface of the ocean, which is where they forage. So they can mostly see the broadband blue scale of the spectrum. And then we also want to look at point number 5 and 4. So those just kind of mean that that whale sharks, when compared to mammals and then also to other sharks, whale sharks deactivate the OPN4 deactivates at a faster rate compared to other sharks because they forage closer to the surface. And so all that kind of combines together in point number 6, which proves the hypothesis that whale sharks have adapted to foraging closer to the surface rather than to light that's deeper in the ocean.
Okay, so when talking about the conclusion, this article was basically just focusing on what they had mentioned previously, the visual pigments found in the genome of a whale, which were the rod RH1, the long wavelength sensitive cone pigments, and the melanopsin. And it should also be noted that although this was only focused on like the genome of two of previously published whale shark genomes, and it didn't focus on other factors such as like the gene expression or the retina inspection. So although it does kind of help go for like provide further insight on how whale sharks have adapted, it doesn't really give us like a definite answer. And it was concluded that the whale shark rod and the cone opsin genes have a visual pigment at around 500 nanometers, and this supports the previous hypothesis that sharks have adapted to their broadband light available at the surface of the water and not the blue shifter light available at depth. And then in regards to the melanopsin, the OPN4, the absorbance spectrum was predicted to be similar to other organisms. I'm pretty sure it was the mice in this case, and then it was at an absorbance maximum at around 480 nanometers. And then the deactivation kinetics. So the deactivation OPN4 was similar to mice, and this was useful when moving from light conditions, so like the surface level light to the lower, and then it just supports their hypothesis that the whale sharks had a duplex retina, and then the deactivation is also tied to mydriasis and pupillary dilation, and the mydriasis is just similar to the dilation, which is just the dilation of the pupil of the eye.
All right, and for future ideas, we found that gene expression could be executed in specific locations. So we could find studies to see exactly where this gene is expressed. And we can kind of understand different factors from that, like what like conditions specifically impact this in more general, like in a more specific manner, and the specific depths that this would occur at. And so we can kind of study their behaviors at a more rate, and we can also like at the rate of expression through the life cycle, because as we know, like the human eye, it kind of worsens with time. We could study this within the gene expression of this gene as well to see how the pigment impacts. We could also see the different behaviors as well. It kind of ties into the first one. We obviously know it impacts foraging. We can also see how it impacts navigation between the whales, and then we can also see how it impacts communication within that as well. And then we can also see longitudinal study to see how areas of high pigment are impacted by human activities such as fishing and tourism, because they are often bycatch products or they are injured by the boats and different types of things like that. And then whale shark hunting especially too, because they are hunted for their meat. And so you can also see how the pigment is expression of that pigment occurs throughout the generations, and kind of see that there's a shift in that with the pigment over time, and how that tells behavioral information as well.